hacker, you could go get a very high-paying job in genetics. So here's a different depiction of that same information. Instead of the machine language of the cell, it's one level up at a functional level of depiction. These boxes are things called genes. Genes encode different molecular functions. So gene number 5, the red gene, that's a gene that encodes a protein called a DNA polymerase. It's just a Xerox copier for DNA. So when the phage needs to copy its own DNA, it makes this protein, it makes more of that. All the blue genes, they make the particle, the shell of the virus itself, and so on. And then you've got a whole bunch of control elements. So this is the architecture of a virus that we found in Nature. It was isolated 60 years ago. You could mix up the program, right? And the short and the long story is, I have no idea what this new program will do, even though it has all the same basic genetic functions. Don't know what it's going to do. So to try and address problems like this, we use computers. Here's a computer simulation of virus infection where we're tracking molecules running along the DNA and reading out the DNA and making proteins and blah, blah, blah. This is open source software. You can go online and find it and run it and simulate at the molecular level any genetic program that you want. The problem with these genetic simulators is that they're not very good, meaning if I were to make a prediction using one of these simulators and then go change my real genetic program, the behavior of my changed program won't usually be what I expect. It'll be something else. So I've spent a lot of time, and apologize, I'm drilling really far down into details here, and I want to do that up front so that I can motivate why I think it's really interesting to hack biology. I've spent a lot of time thinking about why biology is very hard to interact with as an engineer. And so here are the 3 reasons I've come up with. Here's the list of all the functions on